is visible. Okay, 1 minute. I launched the poll. You can tell me. Fine, fine. Let us start the class. Thank you. Hmm. Okay, fine. So, good morning, students. In the today's class, okay. In the today's class, we are going to recall the chapter that is the anatomy of the flowering plant. Okay, let us start the class. Okay. So when we are talking about the anatomy of the flowering plant, here mainly we are going to talk about the internal structures of the plant. Okay, in the morphology of the flowering plant, we mainly study about the external features, right? In the anatomy, we are going to study the internal structures of the plant. Mainly, we can see through the microscope. Okay, fine. Let us study today, guys. First, let us talk about the plant tissue. Okay, can anyone tell me what do you mean by tissue, guys? Tissue. I think it's a simple question. You all know that, but I want you to tell me.
Group group of cells combined to form tissue, ma'am.
Yeah, very good. 1 minute. 1 minute. 1 minute, guys. Okay, fine. When you're talking about the tissue is a group of cells, right? Group of the cell that is having a similar structure and similar function. We call it as a tissue. Here we are going to mainly talk about the plant tissue. Okay. So in the plant tissue, mainly we will talk about the meristematic tissue and the permanent tissue. What do you mean by the meristematic tissue, guys? We will study about that in detail. First, we will classify this all the thing. Okay. In the meristematic tissue means they are not organized, they are not the matured cells, they are the immature cell, and they have the capacity to divide. Okay. In the meristematic tissue, we can see only mainly the primary meristem and the secondary meristem. And in the primary meristem, we can see the apical meristem and the intercalary meristem. And in the secondary meristem, we will see about the vascular cambium and the core cambium. And in the vascular cambium, interfascicular and the intrafascicular, we will see. Okay. We will study that in detail. Just I am telling the classification here. Okay. In the permanent tissue, we can see the simple permanent tissue and the complex permanent tissue. In the simple means they are made up of the similar type of the cell that is having the same structure and having the same function. Okay. Then coming to the complex, they are having the different structure and the different function. Example for the simple permanent tissue are the parenchyma, collenchyma, and the sclerenchyma. Then coming to the complex, they are made up of the different structure and the different functions of the cell. Xylem and the phloem are the examples. This is just about the classification. Let us study in detail. Okay, fine, guys. First, let us talk about the meristematic tissue. Why we are going to call the meristematic tissue means, just imagine in the tissue culture, they will for the culturing they will use the meristematic tissue. Can anyone tell me why meristematic tissue is used for the tissue culture mainly? If anyone know means, can you tell me, guys? Good afternoon, Puneet. Because the meristematic tissue are virus free. Okay, so no disease will become that. So that's why, yeah, disease free plant. Yes, so that's why we will use the meristematic. Okay, okay, because they are rapidly dividing cells. No, that's why the meristematic tissue are the specialized plant tissue. They have the ability to divide throughout the life of the plant. Throughout the life of the plant. So that's why we will call it as the meristematic tissue. Okay, this is not going to be present in the human in the animals, right? It is present only in the plants. Okay, they are contributing to the growth and the development of the plant. Let us talk about the primary meristematic tissue characteristic. These have the ability to grow and divide. They are the these characteristics are the important, guys. In the so many MCQs they have asked these questions. They are the small and immature cell. They are not matured, okay? And isodiametric, round and oval and polygonal shape. And they have the absence of intracellular spaces. They have to divide rapidly, right? So that's why intercellular spaces are not there here. And the cell wall is thin and they are made up of the cellulose, okay? And nucleus is conspicuous, very small. And the cytoplasma is dense here, okay? And vacuole is absent and respiration is high and there's a little reserve food here is there. Okay, fine. Then coming to the primary meristematic tissue, guys. In the primary meristematic tissue, mainly we can see in the root apical meristem and the shoot apical. In the root tip and the shoot tip, you can see the primary meristem. Primary meristem are directly formed from the embryo. Embryo. The zygote will develop, the zygote will be undergo the mitosis to form the embryo, right? From the embryo, the directly primary meristem will be formed. Okay. The primary meristem, you know that it is going to be from the shoot and the root, right? In the root, we can see the root apical meristem. Okay. RAM we will call. And in the shoot, we can see the shoot apical meristem. Okay. In the apical meristem, we can see the shoot and the shoot and the root apical meristem. Okay. Then coming to the, can anyone tell me, guys, in the root, there is a cuticle is present or not? In the root. Root contain cuticle? No, right? Root does not contain the cuticle because cuticle, it helps in the regulate the transpiration, right? So that's why it does not contain cuticle. Remember that. Okay, fine. Root apical meristem and shoot apical meristem. And here you can see, guys, this is the apical meristem. Here it is helping in the growth of the shoot, continuous growth of the shoot. And coming to the intercalary meristem, here it is present near the nodes, okay? In the above the nodes and the below the nodes, you can see the intercalary meristem. This is mainly helpful in the elongation. Okay, it is mainly helpful in the elongation. You can see this intercalary meristem in the grass. You can see that in the grass, right? The grass, the browsing animal will remove the starting part. That is the front starting part. It will be removed by the grazing animal. Then again it will grow, right? Grasses. That is due to the intercalary meristem. Remember that. Okay. Okay. Then coming to the secondary meristem. Secondary meristem usually form from the permanent tissues. Okay. The permanent tissue, again, it will undergo the undifferentiation. Undifferentiation and it will form the secondary meristem or the lateral meristem. Examples are the vascular cambium and the cork cambium. The vascular cambium, there are two types again, guys. The examples are the interfascicular cambium and the intrafascicular and the interfascicular cambium. We will study that in detail. What is that? Okay. Cork cambium, mainly you can see in the bark. Okay, bark. Fine. So this is about the meristematic tissue, guys. These two diagrams are very, very important. They may ask you in the exam the diagrammatic question. Okay. So this is called as the leaf primordia, where the leaf is going to be formed. And here you can see this is the shoot apical meristem. Okay. This is a meristematic zone. And these all are the axillary bud and this is the differentiating vascular tissue. This diagram they may give and they may tell you to label the parts. Okay. So that's why it's important. Usually auxin only. You can see that, yeah. Auxin only, Charan. Okay. Then coming to the next, I think, do you understood what is the meristematic tissue, guys? Do you understood what is the meristematic tissue? If you understood means kindly give me any response, guys. Or if you are having the doubt means I will clearly tell once again. Yes, Puneet, these notes are available. We are giving the link to you. In the link, you can go through this part. Yes. Yes, Jyotika, thank you. Meristematic tissue, just remember the diagrams and the characteristics, guys. Secondary meristem and the primary meristem. That is important there. Okay. Then coming to the then coming to the permanent tissue. Why we are going to call as the permanent tissue means these are permanently differentiated. You can see that meristematic tissue is like the 10th students. Those are not differentiated. Like after completing the 10th, they may opt for the arts also, commerce also, science also, anything, right? But the permanent tissue is like the 12 students. Like you can take 12 student. If you taken the science background, you have to go continue in the science background only. If you taken arts, you have to continue in the arts only, right? Like that, the permanent tissue having the particular structure and they having the particular function to perform. Okay, they will not deviate from that, so that we will call it as the permanent tissue. Okay. Yeah, Puneet. Welcome. Okay, then coming to the permanent tissue, these are the mature one. Okay, we are talking about the mature one, and this will not undergo the any division. Okay, these are non-dividing, and they are formed from the meristematic tissue only, but they are differentiated. Okay, they are formed from the meristematic tissue only, but they are differentiated. Now they have a specific function and they have a specific structure. Okay, so you can see that function like the support, storage, and the transport. These all the function they have particularly. Okay, fine. Then coming to the here, you can see meristematic tissue only that is differentiated to form the permanent tissue. In the permanent tissue, we have a simple tissue, means permanent tissue made up of the cells. All cells are in the similar structure and all are performing the same function. For example, you can take, I have given a one project for you people. For all the 12th student, I have given the project. It's like the simple tissue. All are 12 student only. It is easy to perform the project. But here in the complex tissue, what will happen? The cells is made up of the different kind. The project I have given, I am including all the nine, all the sixth, the seventh, eighth, ninth, 10th, 11th, all the students. It will be difficult, right? Or it is a complex. So that we will call as a complex tissue. All are having the different structure and different function. Okay. But together all are performing the same function. In the simple tissue, the examples are parenchyma, collenchyma, sclerenchyma. And in the complex tissue, examples are xylem and phloem. Remember. Okay. I think you already studied these guys. This is not a difficult for you all. Then coming to the parenchyma cell, collenchyma and sclerenchyma. Here you have to mainly remember the cell wall is made up of and what is the function and this all the thing is important, guys. Okay. Then coming to the parenchyma cell, this parenchyma cells is, you can see in the all the parts of the plant body in the soft parts. Then coming to these are unspecialized cells. These we can see in the every part because having the same, it will do any function kind of thing. And consisting of the thin cell wall, the cellulose, the cell wall is very thin here. Okay. Then coming to the space, you can see the space between the cell. Here you can see in this diagram, there are given very small spaces here. You can see the spaces are there between the cells here. Okay. Remember, only the parenchyma consisting of the intercellular spaces. Okay, they're consisting of the living cell at the maturity. Okay. And these are helpful in the photosynthesis, storage of the food, and the exchange of the gases also. And these are mainly consisting in the water plant, in the aquatic plant that is having the parenchyma that is differentiated into the aerenchyma. Aerenchyma. Okay. So aerenchyma, it is helping in the maintaining the buoyancy that will float, right? Float on the water. There, parenchyma converted into the aerenchyma there. Okay, remember that. And during the photosynthesis, the parenchyma only converted into the chlorenchyma. Remember, chlorenchyma. Okay, fine. Then coming to the cholenchyma, guys. Cholenchyma, these we can see in the petiole because we need, you can hold the petiole like this, right? Petiole will be like this and leaf structure will be like this, right? The petiole is holding the leaf, right? The mechanical strength is given by the petiole, right? In the petiole, we can see the collenchyma. Okay, collenchyma is present in the petiole, leaves, and in the young stem. In the stem, we can see these are specialized cells and consisting of the unequal thin cell wall. Okay, unequally, because in the pit you can see that pectin is present. Okay, you can see here these are unequally distributed. Pectin is unequally distributed. Okay, and you can see here there is a little or no space between the cell, and these are living at the maturity. This is important, guys. Both parenchyma and collenchyma are living at the maturity. Okay, then coming to the main function, this will give the mechanical support. No storage, mechanical support. Okay, then coming to the sclerenchyma, these found in the mature parts of the plant. In the xylem and phloem also you can see the sclerenchyma in the form of the fibers. Have you seen the fiber of the coconut, guys? Coconut fiber you can see, right? In the village side and all, they will use it for the washing their dishes. Have you seen, right? Those all are made up of the sclerenchyma only, fibers. And you can see the fibers are used for the ropes, making the ropes, right? These all are made up of the sclerenchyma only. These are the specialized cell consisting of the thick and rigid cell wall, mainly because lignin is present here. Lignin, okay? The lignin is present here. Pectin is present in collenchyma, lignin in the sclerenchyma. So that's why they are dead. Dead at the maturity. Okay, sclerenchyma is dead at maturity. Remember that. Okay, the main function of this sclerenchyma is protection. They will give the protection because they are very strong. So that's why they will give the protection. You know that we will keep the bodyguard. They will be look like very thin or like the well-builded body. If you become successful someday and you will keep the bodyguards, right? How can you keep the bodyguards, guys? Whether the bodyguard will be very thin, look like very thin? No, right? They'll be like well builded and they will be strong, right? Like that kind. So here they will give the mechanical support, protection, and transportation of the water and the nutrient because they present in the xylem and phloem also. That's why. Okay. Fine. Here you can see the structure, guys. The collenchyma consisting of the pectin here. Okay. And this structure is important of this sclerenchyma. This is the sclerenchyma structure, guys. This is a scleroid. The round-like structure you can see that is a scleroid, and this is a fiber. This fiber is mainly you can see in the coconut fibers and all the things. And here you can see scleroid. Scleroid mainly you can see in the pulp of the guava and pulp of the pear, and the you can see that in the yeah, wherever you can see, guys, in the sapota pulp also you can see this scleroids. In the guava pulp, in the pulp mainly you can see it, and the shells of the nuts. In the shells of the nuts also you can see this scleroid. That is nothing but sclerenchyma. Okay, have you understood this permanent tissue, guys? Have you understood this permanent tissue, simple permanent tissues? Have you understood? If you understood means kindly give me thumbs up, guys. This is important, guys. Scleroid. Scleroid, remember, that is the shells of the nuts. Okay, outer coating of the nuts it is consisting of, and in the pear, okay, pear and also in the sapota and the guava it is present. Okay, fine. Thank you. Okay. Next, go to the complex permanent tissue. We already told this is complex because consisting of the different structure and different function of these cells here. Okay, and but all together performing the same function. Okay, here you can see the xylem and here you can see the phloem mainly. The xylem is consisting the function of the xylem. Xylem is helpful in the transport of the water and minerals, and you can see that xylem is consisting of trachea, vessel, xylem fiber, and xylem parenchyma. Okay. Can anyone tell me, guys, which one is the mainly water conducting cells? Mainly water conducting cells in the xylem. Which component is mainly conducting the water here? Can anyone type? Can anyone type? Which one is the mainly water conducting here? And in the xylem, which one is the dead? Okay, fine. I will tell you. The mainly water conducting are the trachea and vessels, guys. Okay, tracheates and vessels mainly conduct the water here, and xylem fiber mainly give the strength. Okay, it will give the support. And the xylem parenchyma, we already know that it will help in the storage of the food. Okay. And here you can see, guys, trachea and vessel. The trachea and vessels here, the trachea and vessels consisting of the lignin. Okay. Both are consisting of the lignin. Okay. And you can see the xylem vessel here. Xylem vessels are having the perforation or the pits inside. Okay. Fine. Then coming to the gymnosperm, guys. In the gymnosperm, the xylem vessel, okay, xylem vessels is absent here, okay. Xylem vessel is absent in the trachea. Sorry, xylem vessel is absent in the gymnosperm. Only trachea is present, okay. Only trachea is conducting the water in the gymnosperm, okay. Fine. Then coming to the phloem, guys. In the phloem, the sieve tubes are there, and the companion cell, phloem fiber, and the phloem parenchyma. Here you can see, guys, in the xylem, the only living tissue is the xylem parenchyma. Okay, remember that. Okay, xylem vessels also living only. Okay, xylem vessels also living only. Fine. Then coming to the phloem. Phloem is helpful in the transport of the food and the nutrients here. See, consisting of the sieve tube elements, companion cell, phloem fiber, and phloem parenchyma. Okay, and you can see that sieve tube elements. 1 minute. Yeah, 1 minute. Xylem vessels, yeah. Here you can see, guys, the xylem vessels here. The xylem vessel is like a tube-like structure, okay? It will like a cylindrical structure, xylem vessel. And it is having the perforation, okay? This is having, you can see that a pores-like structure is there, opening is like their structure is there, right? The one vessel is attached on the other vessel. One vessel is attached on the another vessel. It is arranged like this, okay? And these all are having the perforation, right? Through that perforation, the water is entering and moving. Okay, in the xylem vessel. Okay, and here you can see the trachea. Tracheas are having the tapering end. Okay, tapering end. These all are having the tapering end, and these all are, you can see that these all are the lignin deposits. Lignin. Okay, fine. Then coming to the phloem, sieve tubes. Sieve tubes are there. The sieve tubes do not have the nucleus. Okay, they don't have the nucleus, so dead at the maturity. Sieve tubes dead at the maturity. This will control by the companion cell. Okay, the companion cell mainly maintain the turgor pressure, maintain the pressure gradient. 1 minute, pressure gradient. Can you tell me, guys, why the pressure gradient is important? Leptome. Puneet, I am not aware of that. I will check once again and I will tell you in the tomorrow's class. Okay, Puneet? But phloem we will call as a bast. Another name for the phloem is the bast. I will check with that leptome. Okay, fine. Companion cell is helpful in the pressure gradient, guys. Pressure gradient. Okay. And sieve tubes are dead at the maturity because they don't have the nucleus. The sieve tubes was controlled by the companion cell. Okay. And the pressure gradient, the companion cell is going to be control the pressure gradient inside the sieve tube because sieve tube is involved in the movement of the food. Here you can see that the sieve tube mainly here. This part is the sieve tube. This sieve tube is involved in the movement of the food, and here you can see that these are the sieve plate. These are called as the sieve plate. Okay. This sieve plate will look like this. Okay. These are having the sieves. How you can see that in the filtering the chai, you can see the filters, right? Like that, in the sieve plate also there will be the filters are there. Okay. Between the one sieve tube to the another sieve tube, there is the presence of the sieve plate will be there. Okay. Fine. Then coming to the phloem fiber, it is also called as the bast fibers. This will be commercially used for the hemp. You can see that hemp, right? Hemp and all the thing will be done, will be used by the phloem fiber. Okay. Then coming to the phloem parenchyma. Phloem parenchyma, mainly it is helpful in the storage of the food. Remember this. Okay. I think you understood about the xylem and the phloem. Now let us talk about the structure here. That is the xylem. This is a vascular bundle which is present. The xylem here you can see, guys. The first formed xylem. The first formed xylem in the plant, we will call it as the protoxylem. Okay, protoxylem will be first formed. It will be smaller in structure. Okay, small. The next formed is called as the metaxylem. That is the bigger in structure. Okay, bigger. Okay. The arrangement of the arrangement of the protoxylem, here you can see it will call as the exarch and the endarch. Arrangement of the protoxylem. Remember. Okay. If the protoxylem is in the periphery, if the protoxylem is at the periphery, we will call that as the exarch. That mainly we can see in the root. Okay. The protoxylem is at the periphery and the metaxylem is at the center. Okay, that is the exarch condition. We can see in the root. But in the endarch condition, the protoxylem are in the center and the metaxylem is at the periphery. Metaxylem we can see at the periphery. This condition we will call as the endarch. Mainly we can see this in the stem. Okay. Remember this point, it is important. It is based on the endarch and exarch based on the protoxylem. Okay, fine. Here you can see the differentiation between the xylem and phloem. Let us once again go through there. Xylem conducts the water and the sap. Okay, and the phloem conducts the organic food. Xylem provides the mechanical strength, and there is no mechanical strength from the phloem. You can see that secondary xylem will provide the mechanical strength, right? Okay. Then xylem is usually found in the deep plant. You can see the outside of the plant. Okay, you can go through this guide. There is no such important things is there here. Okay, let us solve some of the previous year questions, guys. I think if you want me to repeat once again the xylem and phloem, or did you understood the xylem and phloem, guys? Can we go with the questions? Can we go with the questions? If you give the thumbs up, means we can go with the question. This all you all are already studied, right? So that's why I went in the brief. Okay, fine. Okay, guys, let us solve the question. Which of the following simple tissue are commonly found in the fruit walls of the nuts and pulp of the pear? Okay. Once again, guys, 1 minute. Okay. So I will explain the once again the permanent tissue, that is a xylem and phloem, guys. Okay, because this is not in the NCERT, right? That's why some people are getting confusion here. Here, the primary mainly the xylem and phloem consisting of the four cell, guys. Okay, one is the trachea, vessel, xylem fiber, and xylem parenchyma. Okay, the mainly here you can see that this is the xylem. Okay, this is the xylem here. Mainly you can see this part. Okay, this part, this all the part you can see, right? This part is called as a trachea. This is called as a xylem vessels. Okay, xylem vessels mainly consisting of the perforation. Okay, these perforations are there. When the water is entering, right? Water is entering, the water will be entered through this. For example, here it is entered, right? It will move like this. Okay. Here it will move like this. From the root to the all the parts of the plant, it is moving, right? The water like that, it will move upward. Okay. In the xylem vessel and in the trachea will lessly conduct, but mainly is the xylem vessel. Okay, it will move the water in the upward position because till the shoot of the plant, water has to be reached, right? It will move upward. Okay, and the movement of the water is through the capillary movement. We will call as capillary movement. How you suck the water or the juice from the this thing, right? Like the how you will suck, right? So that is like the water is moving in the xylem vessel. Okay. Then the trachea, the both function will be done. That is trachea also do the same and the xylem vessel also do the same. The protection for these both xylem vessel and the tracheas is given by the xylem fiber. Xylem fiber. Okay. It will give the mechanical strength for that, and it should have some storage food, right? That is done by the xylem parenchyma. Xylem parenchyma mainly store the food here. Okay. So this is about the xylem. The xylem composite of this all the four things. Okay. Then coming to the phloem, here also the mainly phloem, it consisting of the four parts, that is the sieve tube element, companion cell, phloem fiber, and the phloem parenchyma. The mainly the food conducting tissue is the sieve tube here, sieve tube element. Okay, but the movement of the food is controlled by the pressure gradient. Pressure gradient. Here also food is moving by the upward and the downward. Okay. So here in the xylem, the movement is unidirectional. 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 The movement is in the unidirectional, but here it is the bidirectional. Bidirectional, right? In the phloem, right? Here what will happen? The food is synthesized from the leaf. It will go to the root also. The root is storing the food that will comes to the other parts of the plants also, right? In the phloem. It is mainly done by the sieve tube. Okay. It will move toward upward and downward. It will move by the sieve tube only. But it is controlled by the companion cell. Okay. The pressure is mainly controlled by the companion cell here. And the mainly support is given by phloem fiber and the phloem parenchyma. Okay. And one more important thing here you have to remember, guys. In the gymnosperm, remember this. In the gymnosperm, they don't have the sieve tube. Okay. In the gymnosperm, they have the sieve cell. Sieve cell is there. Okay. Sieve cell is there instead of the sieve tube. Okay. And companion cell is not there. Instead of the companion cell, albuminous cell is there. Albuminous cell is there in the phloem, in the gymnosperm. Remember this important. Have you understood now? Can you give me thumbs up, guys? Have you understood? Charan, did you understood now? Charan? Okay. Remember the gymnosperm also, guys. That is important. Okay. Fine. Here is the question, guys. So, yeah, you can, I will run the poll. Already I have shown this question to you. I will run the poll. You can go through there. 1 minute. Yeah. I'm launching the poll. Yeah. Very good. Yeah, okay. Okay, guys, fine. So I'm ending the poll here. I'm sharing the result. 64% have given the option A, and 27 has C, and 9% as the D. Guys, see here. Remember the sclerenchyma cell. Okay, sclerenchyma is the correct answer. Scleroids. In that we studied about the in the sclerenchyma we studied about the scleroid and the fiber, right? The scleroid is important. Okay, scleroid is we can see in the fruit walls and the pulp of the pear. Okay, not the fiber. Okay, fiber you can see in the rope and in the coconut things, right? And coming to the parenchyma is having the only the storage and photosynthesis function. Photosynthesis. Okay. Then coming to the collenchyma is present in the only for the mechanical strength. Mechanical strength. Okay. So the correct answer is option A. Okay. So here is the question, guys. Option statement wise question. Statement 1. Parenchyma is living, but collenchyma is a dead tissue. Okay. Then go through the what I have discussed there. Okay. Option statement 2. Gymnosperm lack the xylem vessel, but presence of the xylem vessel is the characteristics of the angiosperm. Okay. Which one statement is correct? Which statement is the correct here? Just go through what I explained it now only. I am running the poll, guys. Again, I am launching. Kindly read once again and then click. Okay. No, can you once again read? Very good, Puneet. Very good, guys. Once again, think. If you given wrong answer, also no problem. Think once again and answer. Okay, I'm getting a different answers here. See here, option A given by the 29, 7%, B 29%, C and 36% D. Okay, guys, see here, which is the correct answer? Option D is the correct answer. See here, parenchyma is living, but collenchyma is a dead tissue. Have you seen that? Only sclerenchyma is the dead tissue, guys. Remember. Okay. Sclerenchyma is living. Collenchyma is the living. Okay. Only sclerenchyma. Only sclerenchyma. Kindly make down this. Okay. Only sclerenchyma is the dead. Okay. In the permanent simple, parenchyma and collenchyma both are living. Okay. Then coming to the gymnosperm, second statement. That's why it is wrong. Okay. Gymnosperm lack the xylem vessel. Now only I told, yes or no? Yes. They lack the xylem vessel. They have only the tracheates. Tracheates. They have only the tracheates for the water conducting, right? And the presence of xylem vessel is a characteristic of angiosperm. Yes. Xylem vessel is present only in the angiosperm. Okay. Till pteridophytes, only vascular bundle started in the pteridophytes. Pteridophytes is also having the tracheates only. Okay. Fine. The correct answer is option D. Statement 1 is false and statement 2 is true. Okay. Fine. Here also, the end arc and X arc are the terms often used for describing the position of secondary xylem in the plant body. Statement 2, XR condition is the most common feature of the root system. Can you answer to this question, guys? Which one is the correct answer? Yeah, you can answer to this. I'm relaunching the poll. Okay, I'm ending the poll, sharing the result, guys. So see here, 25% given A, 38C and 38D. Okay, no problem. See here, guys. We have seen the two types, right? One is the exarch and the endarch based on the xylem, right? Protoxylem means that is a smaller, and the metaxylem is, see here, metaxylem is larger, protoxylem is smaller. Proto is the first formed and meta is second formed, right? So here you can see if the protoxylem is the center, and you can see that, okay, you can see that protoxylem is the center and the metaxylem is the periphery. Okay, this is called as the end arc condition. Okay, if the protoxylem is at the outside and metaxylem is at the, sorry, metaxylem is at the center. Metaxylem is at the center, protoxylem at the periphery. This is called as the X arc. X arc, right, guys? X arc. In the X arc, it is in the periphery. In the end arc, it is in the center. Here you can see end arc and the X arc are the term used after for describing the position of secondary xylem in the plant body. No, it is describing the protoxylem. Okay. Then coming to the statement 2, XR condition is the most common feature of the root system. Yes, we studied, right? XR, XR, we studied that. That is important, that is present in the root. And endarc we studied, that is present in the shoot. Sorry, in the stem. 1 minute. Endarc mainly we can see in the stem. Okay, fine. Here, so that's why the correct answer is C, guys. See here. Statement 1 is wrong, right? Statement 2 is true. This is a wrong and the statement 2 is the correct. XR condition we can see only in the root. Okay. Fine. Let us go to the next question. Interfascicular cambium is present between. I'm running the poll. Okay, secondary xylem and secondary phloem. Option B, primary xylem and primary phloem. Option C, pericycle and endodermis. Option D, vascular bundle. Okay, I'm ending the poll, sharing the result. 14% given A and 43% C and 43% D. Let us study about this, guys. You can see here the interfascicular cambium. You can see this, guys. This is the conjoint system. Here you can see this is the phloem. Okay, this is the phloem and this is the xylem. Okay, this is xylem. Between this, the cambium is there. That is called as the intrafasciculum. Intra, because it is inside, right? Intrafascicular. Okay, intrafascicular cambium. Okay, and here you can see there are vascular bundles are present. Okay, so here the medullary race will be there. Here it is going to be form the ring-like structure, right? That ring-like structure we are going to call as the interfascicular cambium. Interfascicular cambium. Okay. That going to be present between, these are a vascular bundle, right? These all are the vascular bundle. The interfascicular cambium present between the vascular bundle. Here it is going to be present. That is between. Okay. Intra means inside. Between means inter. Okay. The correct answer is option D. Okay. The initiation of the lateral roots and the vascular cambium during the secondary growth takes place in where? Option A, pericycle. Option B, epiblemma. Option C, cortex. And option D, endodermis. I'm launching the poll. You can answer me, guys. Okay, I'm ending the poll. See here. The correct answer is given by none of this. None of one. Okay. So let us answer to this question, guys. Okay. The initiation of the lateral root and the vascular cambium mainly present in the pericycle. Okay. Option A is the correct answer. Okay. So girdling experiment was performed by the plant physiologist to identify the plant tissue through which. Okay. So you can answer to this question if you know means. Okay. Option A, water is transported. Option B, food is transported. Option C, for both water and food transportation. Option D, osmosis is observed. Very good. Okay. Okay, guys, I'm ending the poll. I'm sharing the result. 13% given A, 25% B, 38% C, and D is by the 25%. Okay. See here, guys. So girdling experiment is mainly performed for the how the food is going to be transported. What they have did in the girdling experiment is they have removed the bark. In the center you can see there is a tree. In the center they have removed the outer part, that is the phloem and the bark. The secondary phloem and the bark has been removed, and then they observed that the accumulation of the food is happened here, and the root is started to because the root is started to die because the food is not transporting to the root. That's why, okay. The girdling experiment is done by removing the bark and phloem. Okay. So you know that phloem is mainly the important for the food transportation. When we remove the phloem, the food transportation has been not taken place. Because of that, you can see the food is not went to the root. The root is died. Eventually, the plant will also die. Okay. Fine. So let us go to the main part, guys. These are the tissue system. Tissue system, there are the different tissue systems are there. One is the epidermal tissue system, ground tissue system, and the vascular tissue system. Okay. First, we will go to the epidermal tissue system. As we know that epidermal means outside part. Okay. The outside part or the external part we will call as the epidermal tissue system. Here mainly you can see that epidermis will be single layer. Okay. It will be single layer, okay, and it is consisting of the parenchyma. Mainly the epidermis consisting of the parenchyma only. Remember, we studied the permanent tissue, right? Epidermal tissue mainly consisting of the parenchyma. In the leaf, we can see the cuticle. The cuticle is helps in the regulating the transpiration. Then coming to the epidermal tissue consisting of the stomata also. Stomata regulate the transpiration. I think you know what is the transpiration, right? The removal of the water in the form of the vapor, right? That is called as the transpiration. Here you can see the structure of the stomata, guys. Okay. This is the dicotyledon stomata and this is the monocotyledon. This is the monocotyledon stomata. Here you can see the shape, guys. This structure is very, very important. Here you can see that the pore is there. That is called as the stomatal pore. The stomatal pore is guarded by the guard cells. Guard cells are there here. Okay. These guard cells. These guard cells are inner side. Inner side is thickened here. Inner side is the thick. Remember this. Inner is the thick here and outside is the thin. Okay. Outside is the thin. Okay. Outside is the thin here. Okay. And this having the subsidiary cell surrounding the guard cell and the epidermal cell outside. Okay. And you have to remember The stomatal apparatus. The stomatal apparatus consisting of the stomatal core, guard cells. Okay, fine. So this guard cell mainly important in the maintaining the closing and opening of the stomata. Okay, when you think the guard cells are the turgid. When the guard cells are the turgid, the water is there inside that. Then the stomata will be open. When the water is not there, when the guard cells will be flaccid, the stomata will be closed. Okay, remember that. Then coming to the monocotyledon, in the monocotyledon, the guard cells are the dumbbell shape. Dumbbell shape. Okay. Okay. Then coming to the epidermal appendages, guys. Epidermal appendages means it is present on the epidermis. Okay. Epidermis. The example, the epidermis of the root that is consisting of the unicellular hair, root hair. Okay. And in the stem mainly consisting of the trichomes in the stem. Okay. The trichomes are the structure. Those all are multicellular in nature. They also have the function that is secretion. They will secrete some of the mucus like or the carbohydrate things also. Okay, fine. Then coming to the ground tissue after the epidermis till the vascular bundle. Okay, except the epidermis and the vascular bundle, all are called as a ground tissue only. Okay, in the ground tissue we can see mainly the parenchyma, collenchyma, and the sclerenchyma. In the leaf, the ground tissue we name as the mesophyll. Okay, remember. Then coming to the vascular system mainly consisting of the xylem and the phloem. Okay. And here you can see guys, conjoined open, conjoined close, and a radial is there. It is nothing but the arrangement of the xylem and phloem. How it is going to be arranged the xylem and phloem in the pericycle. Pericycle. In the pericycle, the vascular bundle is going to be arranged. Okay. How it is going to be arranged? Here you can see that this is the xylem, this is the phloem, and this is the xylem inside. These are arranged in the radial structure, radial, in the alternative manner, right? Here you can see the phloem, phloem, xylem. This is in the alternative manner, alternative, right? And this structure is called as a radial. Okay, this arrangement is called as a radial. For example, if I tell the student to sit like the in the round means one boy, one girl. If you sit like this means the xylem and phloem is sitting like that. Okay, so that we will call as the a radial, and this alternative manner, that is a radial. Mainly we can see that in the root. In the root we can see the arrangement of the xylem and the phloem as the radial. Okay. Then coming to the conjoint open means between the phloem and the xylem, there is a cambium tissue is there. Cambium is there means why we are called as a conjoint open. Conjoint means xylem and phloem are arranged in the same axis. In the same axis. For example, this is the same axis. Okay. This phloem is there and xylem is there. So that is called as the same axis. That is conjoint. Open means what? Open means the cambium is present, means they are open to the secondary growth. Okay, means they can increase their girth. They are open for the secondary growth means there is a presence of cambium is there. Okay, and that's why this conjoint open is present in the dicotyledon. Okay, the cambium is present between the xylem and phloem. Whereas in the conjoint closed, they they are not undergoing the secondary growth. So that's why we call as a closed. Between the phloem and xylem, there is no cambium is present. That is called as conjoint closed. Okay, fine. This is about the tissue system, guys. Let us talk about the root structure. Kindly see this diagram, and the labeling is important, guys. The epidermis, okay, here you can see the epidermis is bearing the unicellular root hairs, okay, unicellular root hairs, and these all are called as the cortex. The cortex mainly consisting of parenchyma. Okay, cortex. You can see this, the space are there, right? This is the cortex. After the cortex, there is the endodermis is there. This endodermis is also made up of the parenchyma only. Okay, parenchyma only, but in the endodermis, the parenchyma don't have any space. Okay, they are tightly arranged in the endodermis. Okay, and you can see that after the endodermis, there is a pericycle is there. In the pericycle, you can see the parenchyma only. Okay, in the pericycle also parenchyma only you can see here in the root. Okay, parenchyma is there, and in the pericycle, the xylem and phloem bundles are there. That is the vascular bundle are there. Here you can see, guys, the Protoxylem is outside. Protoxylem, smaller one, is outside, and the metaxylem is inside, right? So this condition we are called as the XR condition, XR condition. Okay. And here you can see the pith is very small here. Small here. Okay. Small. And the vascular bundle, you can see only four. Only four vascular bundle you can see, that is in the dicotyledon root. This is how you can differentiate between the dicotyledon root and the monocotyledon root. Okay. And the pith is there. In the pith also there is the parenchyma cells will be there. Okay. And here you can see that the uh, the cells which are between the vascular bundles, okay, that is called as the medullary parenchyma. These are also parenchyma only. Medullary parenchyma cells. Okay. So this is about the dicotyledon root. What you have to remember in the dicotyledon root is mainly four vascular bundle is there and the XR condition is there. Okay. Then coming to the monocotyledon root, the same thing, guys. Outer side you can see that epidermis and cortex is made up of the parenchyma only, and endodermis is there, and the pericycle and vascular bundle is there. But you can see that protoxylem is the outside and the metaxylem is inside. Okay, the same thing. Same thing here. Protoxylem is outside, metaxylem is inside. It is called as the X-arc. In the monocotyledons also you can see the root is the XR condition only. But only different thing is here you can see the polyarch. Polyarch. What do you mean by polyarch, guys? Here more than six vascular bundle are there. Okay, more than six vascular bundle are there. Here you can see 1, 2, 3, 4, 5, 6, 7. More is there, right? More vascular bundle is there. Okay, xylem bundles. Okay, and the arrangement here is radial type. Okay, no conjoint, radial type is there. Okay, fine. Both are same only, though only differentiate is the polyarch condition in the monocotyledon. Then coming to the stem, guys. In the stem, if you take the stem cross section in the dicotyledon, mainly you can see the epidermis. Epidermis is bearing the you can see the epidermis is bearing the trichomes. Okay, trichomes you can see, and here you can see the hypodermis. Hypodermis mainly consisting of the collenchyma. Okay. And after the hypodermis, there is the dermis is the epidermis is there. Epidermis consisting of the parenchyma. Epidermis consisting of the parenchyma, then the endodermis, then pericycle. The endodermis mainly consisting of the starch granules. Remember, guys. The endodermis the endodermis mainly consisting of the starch granules, which are rich in the starch granules. Okay. And the pericycle here you can see, pericycle is mainly consisting of the sclerenchyma. And after that pericycle, you can see the phloem and the xylem arrangement in the form of conjoint open. Okay, vascular cambium is there. Here you can see the phloem is there, xylem is there, and the between there is the vascular cambium is there. Okay, so that's why we are calling as the conjoint open. You can go through this. Okay, these all are the trichome, and after the trichome we can see the hypodermis. Then we can see the in the hypodermis collenchyma is there. Then below the hypodermis is the epidermis. In the epidermis we can see the parenchyma. Then after that we have the endodermis. Endodermis are rich in the starch granules, and after the endodermis you can see the pericycle mainly consisting of the sclerenchyma. Okay, and the vascular bundles are arrangement are the conjoint, and the pith is there. Okay, pith is large. And the presence of the vascular bundle, here you can see this is a dicotyledon that is open, and here you can see in the dicotyledon secondary growth is there. Okay, secondary growth you can see. Okay, then coming to the monocotyledon stem, guys. So here you can mainly differentiate the epidermis and the hypodermis. Hypodermis mainly consisting of, here you cannot see the trichomes. Okay, the hypodermis mainly consisting of the sclerenchyma. Hypodermis mainly consisting of the sclerenchyma here, and the vascular bundles are scattered. Here you can see these all are scattering, right? Vascular bundles, and these vascular bundles mainly consisting of the a bundle sheet. Okay, bundle sheet that is consisting of the sclerenchyma. Bundle sheet is covered by the sclerenchyma. Okay. And the parenchyma is absent in the monocotyledon stem. Remember. Only thing you have to remember here is vascular bundle is scattered and the hypodermis is made up of the sclerenchyma and the bundle sheet cell is there. That's it. Okay. Then coming to the leaf structure, guys. Leaf structure is very, very important here. The dorsiventral leaves, you can see in the dicotyledon root. Sorry, dicotyledon leaves, you can see the dorsiventral leaves. Okay. So here what will happen? Let us go to the structure here. The adaxial epidermis. Adaxial means dorsal. Okay. This is the dorsal surface and this is the ventral. Abaxial. A is there, right? A means you can see that is the ventral. Okay. D here is there. So that is dorsal. Okay. In the adaxial or in the dorsal surface, we can see the parenchyma that is differentiated into the pallicidae, mesophile, pallicidae parenchyma. Okay. So that are elongated parenchyma we can see in the dorsal side. And also you can see that stomata is less here. Stomata is less here in the dorsal surface. Okay. Then coming to the ventral side, the stomata is more stomata is more here, more over here. Okay. And in the ventral surface you can see the mesophile cell, that is the spongy mesophile. Okay. Spongy, and they are having the gap, they are having the air cavities, whereas in the pallicidae there is no air cavities are present. Okay. And the vascular bundles are present in the rib, in the midribs of the leaves we can see the vascular bundle. So this is about the dorsiventral leaf, that is in the dicotyledon. Then coming to the monocotyledon leaf, we call it as the isobilateral leaf. Okay, we cannot differentiate the ventral surface and the dorsal surface there mainly. So that we can see in the monocotyledon leaf. Here you can see the equal. Okay, equally we can see the stomata. Stomata we can see equally. Okay. So here you can see the adaxial epidermis and the abaxial epidermis. What you have to see here is there is no differentiation of the pallicidae parenchyma and the spongy parenchyma here. Okay. So parenchyma is not differentiated into the pallicidae and the spongy parenchyma here. Here you can see some of the, in the adaxial surface or in the dorsal surface, you can see some of the parenchyma is uncolored. They become the white color. And here they mainly help in the turgidity. So that cells we call as the bulliform cell. They are present in the dorsal surface, bulliform dorsal cell. When the bulliform cell is turgid, they will expose the leaf. Okay. They will expose the leaves when they are flaccid. When they are flaccid, have you seen the leaves will be curl out, right? They will curl. When they are flaccid, they will curl out the leaf. Okay. That we can see mainly in the bulliform. Bulliforms are the colorless. Okay. Colorless, they form only in the dorsal surface. This is important, guys. That's it. So here is some question, guys. Let us solve this. I think we run out of the time, but I will give you this PPT. You can go through the next slides. Okay. Fine. Let us solve this question, guys. Read the following statement about the vascular bundle. In the root, xylem and phloem in the vascular bundle are arranged in the alternative manner along the different radii. Alternatively, they are arranged. Okay. The conjoint closed vascular bundle do not possess the cambium. They are closed. Okay. Then see, in the open vascular bundles, cambium is present between xylem and phloem. Option D, vascular bundles of the dicotyledon stem possess the endark protoxylem. And in the option E, in the monocotyledon root, usually there are more than 6 xylem bundles are present. Polyar condition in the monocotyledon root. Okay. So now you can choose the correct statement. The correct statement. This question is asked in the NEET 2022. You have to tell me the correct option from this statement. Okay, I will launch the poll, guys. Yes, Maya, you will get the link of the notes in the group. Punit Kumar, age of the tree will be estimated by the spring path. That is the ring system. I will tell you. 1 minute. Have you understood this question? I will tell you, Puneet. Give me a 2 minute. By the cambial ring, we will calculate the age of the tree. Cambial ring. I'll launch the poll, guys. You can answer me in the poll. Cambial ring. Have given all the thing in this PPT. You can go through that. Okay. I'm ending the poll. Here is a twist is there, guys, for you all. What is the twist is all the options are correct actually. Okay. But in the exam, they are given the option B is the correct answer, but all the statement here are correct only. In the root, the phloem and xylem bundle are arranged in the alternative manner. That is correct. That is a radial, right? That is correct. Conjoined closed vascular bundle do not possess the cambium. That is correct. Conjoined closed those does not contain the cambium. That is also correct. In the open vascular bundle, cambium is present. Yes, they are open to the secondary growth. That is correct. The vascular bundle of the dicotyledon stem possesses the end arc. Yes, stem contain the end arc, root contain the X arc. That is correct. The monocotyledon root usually are more than 6. Yes, correct. That is the polyarch. All are correct only here. Okay, fine. These all are the question, guys. This is a secondary growth. You can go through this slide. And this is the annual ring. Now only you asked Puneet, right? So the according to the annual ring, based on the annual ring, we can calculate the age of the tree. Okay. So in the annual ring, it is consisting of the autumn wood. That is the two rings will be there. One is formed in the autumn and one is formed in the spring. Right. In the spring wood will be in the light in color. Autumn wood will be in the dark in color. We are counting both the dark color and the light color together. We are calling as the annual ring. Okay. So based on the annual ring, we are calculating the age. Okay. And you can see that these all are the secondary xylem only. Okay. Secondary xylem only. The secondary xylem started to deposit here. In the center, it is started to deposit the uh, secondary metabolites like the tannin and all the things. So that become dead. It is not going to be transport the water and the minerals. The heartwood that is resistant to the insects. Okay. The heartwood will be resistant to the insects. And here you can see the sapwood that will be light in color. That is possess the secondary xylem that is going to be transport actually the water and minerals. Okay. You can go through this and this PPT also. And some questions also there. You can go through this guys. Okay. I'm launching the MPS poll. You can give me your feedback. 1 minute. Yeah, I will launch the NPS poll guys. If you are having any question, kindly ask me. No, here they are given in the different radipa, not in the same radi. Different radi in the question they are given different radi. In the different radius only there, no? Shruti, they are in the different radius, not in the same radi. Okay, guys, you can go through this. Actually, this chapter is easy only, but some of the concept has been deleted, no? So that's why it took the time, actually. Okay, you can go through this with this PPT I have given clearly. Okay, I will upload this. We will give the link also. Okay, fine, guys. Thank you so much. If you have any questions or any difficulties, kindly contact me. Okay, thank you so much, guys. If anybody is having the question, please stay in the call and you can ask me the question, guys. Thank you, Nandita.